I'm Tandy Warnow. I did my PhD in mathematics at Berkeley a long time ago. I've been at CGSI now for 3 years, and I work mainly on phylogenetic estimation, also on multiple sequence alignment, also now on clustering, also on microbiome analysis, and also in historical linguistics. So if you're interested in one of the topics that is not, if you're interested in any of these topics, come and see me during some coffee break. Okay, so this is the tutorial on phylogenomic tree estimation, and I'm actually going to cover a bit more than just that one topic, but phylogenomics is basically the combination of whole genomes and phylogeny estimation. So phylogeny estimation is estimating evolutionary histories, and you can do this as a tree or you can do it as a phylogenetic network. Today I'm not going to be talking about the phylogenetic part. I talked a bit about that yesterday. So there's this complex pipeline for doing a phylogeny on a bunch of species. You're figuring out which species you want to look at, which genes you're going to look at. You're screening your data. You're figuring out orthology potentially. You're getting multiple sequence alignments. You're getting gene trees. You're combining the alignments or combining the gene trees. If you combine the alignments, you might then run a tree estimation method on the combined alignment. And at the end of this process, you have a tree. You then want to get branch support, you want to get dates, you want to do various things so you can answer biology. The point is that this is a complex pipeline, and mistakes early on lead to mistakes later. And understanding where the mistakes are coming from and having some sense of what you can have some confidence in is a big part of a phylogenomic analysis. What I work on, as I said, is multiple sequence alignment, gene tree estimation, and species tree estimation, and also to some extent phylogenetic network estimation. Today, I'm only going to be talking about the tree estimation parts. So tree estimation for a single gene, or tree estimation for a multi-locus data set. And they're not exactly the same thing, but I'm going to start with the gene tree so you have the concepts. Okay, so a lot of this is based upon my experience with working with different international projects. The first one I worked on was the Avian Phylogenomics project, which was quite a while ago. We had roughly 48 birds, but we had whole genomes. And when we did this analysis, which took, in fact, like 3 years to do this project, we ended up with a multi-locus species tree that took, well, we used maximum likelihood, so RAXML. It took 250 CPU years to do this analysis. So that's a lot of time for 48 species. And I just want you to have a context that this can be computationally intensive. There was also a lot of gene tree heterogeneity that was consistent with incomplete lineage sorting. So another project was the plant transcriptome project, 1KP. Somewhat more species, but we had even more heterogeneity. And the big issue here was multicopy gene families. So multicopy because genes are evolving with duplications. And if you don't have orthology figured out in advance, you have multicopy gene families. That makes for extremely large alignment problems and complicated species tree estimation. And again, gene tree heterogeneity that was consistent with incomplete lineage sorting. I keep mentioning incomplete lineage sorting because that's a big part of what this talk is going to be about. So large phylogenomic data sets are difficult, but there's two types of large. One is the number of loci, which is to say genomic regions. I also just refer to them as genes. Genes doesn't mean protein coding. It just means a region in the genome. So the number of loci is one. The number of leaves in the tree that you're constructing, that could be species or individuals, is another. Two different dimensions for large. And you can have missing data, you can have heterogeneity in the evolutionary process, and all of this makes for complicated statistical estimation and challenges in using your software, especially since a lot of the methods people want to use are based upon maximum likelihood or Bayesian estimation, and one way or the other, they're computationally intensive. So this talk, I'm going to start with gene tree estimation, then I'm going to do species tree estimation. Then, if I have time, I'll tell you some new things we're doing in how to improve scalability, and then we'll have some discussion of open problems. So gene tree estimation, the basic concept here is you have a single genomic region, it evolves down a single tree. This is the classical problem in phylogenetics. So you have a sequence at the root, and over time it's evolving. And I'm just highlighting in red where things change. And so it evolves over time, and you have speciation events, and you end up with the sequences at the leaves. And from the sequences at the leaves, you want to reconstruct the tree. 2 points. This cartoon has no insertions and deletions. It's just one sequence evolving with substitutions. Okay? Second thing is the statistical models of evolution are time reversible. And when they're time reversible, you can't figure out the root. So you will consider yourself to be happy if you get the correct unrooted tree. Okay? So that's the objective, to get the correct unrooted tree from the sequences. If you want to then root the tree, you typically use an outgroup. So you take, for example, you've got a lot of birds, but now you've got a crocodile. So you rooted a crocodile. Okay? So rooting the tree is usually done with an outgroup. You can do it in other ways as well. To make this a statistical problem and to be able to look at its properties, you have to have a formal model of sequence evolution. And so there are lots of them. This is the simplest one. It's called Jukes-Kanner. And what you have is each of the positions in the sequence evolve independently of each other and under the same process. And every edge has a substitution probability. And if you change on the edge, you change to one of the other nucleotides with equal probability. Obviously, this is extremely simple. You can make it more complex by having a more complex substitution matrix. You can have race across sites. You can do various things here. And almost every change you make doesn't change the mathematics. You can still do statistically consistent estimation under the model. If you make the changes dramatic enough, then you can't. So I'm going to be sticking with the ones where you can do statistical estimation, such as the generalized time reversible model. Okay, so it's a statistical estimation problem. There are many methods. Here are 4 categories that are pretty common. Maximum likelihood, which is to say you know something about the model. For example, you know it's Jukes-Kanner. You want to find the model tree that has the highest probability of generating the data. Okay? It's an NP-hard optimization problem, very cleanly stated. Bayesian estimation is basically a random walk in model tree space, and after a while you've converged to the stationary distribution and you can sample. Distance-based methods are where you calculate a pairwise distance between every pair of sequences, making that calculation under the model, and then you use the distances to construct a tree. Finally, maximum parsimony, which is saying, I just want to minimize the total number of changes. 4 basic categories of methods. They're popular, each of these is popular in some group of researchers. Some people love distance-based, some people love parsimony, some people love maximum likelihood, some people love Bayesian. You know, that's really just, it's like group by group, they have the things they like to use. Because almost everything is NP-hard, there's lots of heuristics. Okay, so when we evaluate a method, we want to look at its statistical properties. Is it statistically consistent? How much data does it need to be accurate with high probability? That's a sample complexity problem. Then there's computational performance, runtime, and memory usage. Then there's accuracy, by which I mean on data. So on simulated data or on real data. The problem with real data is you don't usually know the truth. The problem with simulated data is it's not necessarily realistic. But you basically look at both. Okay? So these are the types of things you look at. So what do we mean by quantifying accuracy or error? If you have a simulation, you have the true tree up here. You have sequences that have evolved down the tree. You then estimate a tree, and I'm marking the tree with branches either as FP or FN. FP means false positive. This edge splits 2 leaves from 3, and that particular split does not exist in the model tree. Okay? So it is a false positive. Now this edge, which is S1 and S2 from the remaining, is also in the model tree. It's right here. So that's a true positive. So you can look at the number of true positives, the number of false positives, number of false negatives. If we talk about error rate, you're only looking at the number of internal edges that are false positives or false negatives. When both trees are fully resolved, it's the same error rate. Okay? So a 50% error rate means half of the edges are wrong. Statistical consistency under the model says as the data goes to infinity, your error should go to 0, and you need to be able to prove this, not just do a simulation. And I'm trying to emphasize this because all too often you'll see papers where they say we demonstrate statistical consistency in this simulation, or we show it's not consistent in this simulation. You can't do either one, okay? You may just not have enough data to establish inconsistency or consistency. And in any event, a simulation doesn't prove anything. It's just a simulation. But it's an important perspective because it's what happens on finite data. Consistency is not about finite data. It's what happens in the limit. OK, so you might say consistency, if it's only what happens in the limit, who cares? So that's why we also think about other things like performance on data and sample complexity. So here's the rough top-level thing. Distance-based methods can be statistically consistent if you do the calculations correctly. They're often polynomial-time. They're fast in practice. Maximum parsimony, NP-hard, there are good heuristics, but it can be provably statistically misleading. It can converge to the wrong tree. This is something that Felsenstein showed years ago. Maximum likelihood, statistically consistent if solved exactly, but who can solve an NP-hard problem exactly? And then finally, Bayesian methods, they're statistically consistent, but they have to converge. Convergence for a Bayesian method can take a very long time. So you have these methods, some of them are fast, some of them are slow. Guarantees are always in context. You know, solving something exactly, fine. Again, it has to be under the model. So this is sort of basic stuff that almost everyone in phylogenetics knows. Some of the stuff I'm going to be showing you, not everyone knows. Sample complexity. How much data do you need to recover the tree with probability greater than 1 minus epsilon? So under the Jukes-Kanner model, every edge has a substitution probability. If you say the smallest substitution probability is F and the largest is G, then if F is very small, you need a long sequences to even get a change on the edge. And if G is very large, things can look random. So you really want a sample complexity that depends upon F and G. But if you just say F is not going to be, it's going to be no smaller than this, G is not going to be any larger than that, then we can talk about the sample complexity now as a function of N and the method. N is the number of leaves. And there's this concept of absolute fast converging. Does your method have a polynomial in which it will be guaranteed to recover the tree with probability at least 1 minus epsilon given sequences of that length, without knowing F and G, okay? It can't be told F and G, it just has to work anyway. And the first fast converging methods were developed not too long ago, but visually it looks like this. Sample complexity says how quickly are you going to 0. Statistical consistency just means you eventually go to 0. So we're also interested in methods that are exponentially converging. So they need something like 2 to the n, or something like that, okay? So distance-based methods are the easiest methods to analyze, as it turns out. This first step, you're estimating pairwise distances. And those pairwise distances, if you correct them under the model, will converge to something called an additive matrix. An additive matrix is a matrix that equals path distances in the tree. So you're looking at the edge weights. And the edge weights here are the expected number of changes. And you can see that this matrix exactly fits this tree. Every pair of leaves, you get exactly the same number, okay? As soon as you have an additive matrix, you will get the true tree using many methods. If you're close enough, you're also guaranteed to get the true tree. And therefore, you have statistical consistency. But what about sample complexity? It turns out that neighbor joining needs distances to be exponential in the maximum leaf-to-leaf distance to have that guarantee. Exponential in the maximum distance. That still gives statistical consistency, but it gives you an insight that its accuracy may degrade with high diameter. Okay? Neighbor joining, very, very basic method for phylogeny estimation. So here's a simulation study that I did with Luai Nakla years ago. Luai Nakla is a former student of mine. And this is as you take one fixed value for F and G, but now you're taking bigger and bigger and bigger trees. If you take bigger and bigger trees, the diameter goes up, even though F and G are not changing. And as the diameter goes up with more and more leaves, the error goes up. Remember, this is the proportion of the edges you're getting wrong. So if you only have, I don't know, 100 leaves, it's fine. But at 400, the error rate is 20%. At 800, it's like 30-something%. Here it's above 40%. Error rate is going up. It can't go higher than 100. Okay, so neighbor joining has this problem. It's statistically consistent, but exponentially converging. So as I said, these absolute fast converging methods, there's a long history here. This is stuff that probably many of you don't know. It goes back to 1997 with Peter Erdős, not Paul. So my Erdős number is 2, not 1. Goes all the way back to 1997. We've been developing methods. Other people have also been developing methods looking at sample complexity and phylogeny estimation. But there's this nice general technique called discovering, okay? DCM1, it's a nice idea for how to improve sample complexity. So the idea is here. You have a data set and a method. In the first stage, you're going to break the data set into subsets. And how you break into subsets is by saying how large a diameter you're willing to consider. So you're using chordal graph theory to do this decomposition. You get a bunch of subsets, you get trees on the subsets, and then you combine them. So in this first stage, you're going to compute a collection of trees, one for each way of bounding the diameter. In the second stage, you score the trees, and you pick the tree that has the best score using a quartet-based technique. So this two-stage technique has this really nice theorem, which is that if you start with a statistically consistent method, you maintain statistical consistency. But better, if you start with something exponentially converging, it becomes absolute fast converging. So it's a generic technique for taking a method and making it a new method. And this new method is DCM-boosted version of the method. So here, remember neighbor joining? So how does DCM neighbor joining look? That's how it looks. So nice, right? Nice theory, absolute fast converging, polynomial time, and error rate's not going up anymore. It'll eventually go up, but it hasn't gone up yet. So that's an idea for what you can do about sample complexity and how it's an important consideration. So are we done? You know, is that good enough for gene trees? And unfortunately not. Because it turns out that even these ways of improving distance-based methods are not as good as maximum likelihood. Maximum likelihood heuristics, not exact solution, just heuristics, they're good. And when you run them on simulated data, they're good. Now they can take more time, but they give really good accuracy. And RAxML was always at least as good and typically much better than any of these other methods. So always the benchmark is, can you improve upon a good maximum likelihood code? It's not easy. That's just out there. Just bear that in mind. So DCM neighbor joining versus maximum likelihood is better. So again, back to this sample, distance-based methods, good, statistically consistent, polynomial time, but their accuracy degrades with evolutionary diameter. That's number one. Maximum parsimony, I would just say it's controversial. There are times when it's good, there are times when it's not good, and it's still controversial. Maximum likelihood is very accurate in practice, but truth is large data sets can be unbearably hard. Remember the avian project, 48 birds, 250 CPU years, okay? And then Bayesian methods, highly accurate in practice when you can run them, but they have to converge. So that's my sample update on what you can do with gene trees. I'm switching now to species trees, which was the topic of this tutorial, but this is the context. You want to remember the context, okay? So we're interested in species trees. Human, chimp, gorilla, orangutan is an example. Here we're saying human and chimp are more closely related. The problem is gene tree discordance. Gene tree discordance says that across the genome you get different trees. You take one gene, you get one tree. Take another gene, you get another tree. And what I mean by gene, again, is just a region in the genome where that region evolves down a single tree. Okay? So different trees in different regions of the genome. It's due to multiple different causes. The most well-studied one is incomplete lineage sorting. Incomplete lineage sorting exists in almost all data sets. Gene duplication and loss is another one, which is very, very endemic in plants. Horizontal gene transfer is another one, very endemic in bacteria. So you get lots of different causes for discordance. Okay? Incomplete linear sorting, as I said, is the one that has the most math about it. So the multi-species coalescent is a model for how you get incomplete linear sorting. And imagine you have a species tree, and you evolve genes within the species tree. Now you have different gene trees. Now these gene trees evolve sequences, and you get sequences. And now if you have the alignments of the sequences, this is what you have as your input, and you want to get the tree, right, that generated the data. So one thing you can do is just concatenate the alignments and run maximum likelihood. It's a very standard technique. It's called concatenation. It's very computationally intensive. Remember avian. So the mathematical model, though, for the multi-species coalescent allows us to prove theorems about methods and get statistically consistent methods. So this traditional approach of concatenation is not statistically consistent when you are working with the multi-species coalescent. It can be positively misleading. But in simulations, it's typically not bad. And I'm going to show you the result a little bit later. Sometimes it's very good, sometimes it's bad. When you should use it and when you should not use it is something you have to develop some understanding about the conditions where methods do well. But what can you do that is statistically consistent is estimate the gene trees, and then from the gene trees, estimate the species tree. So that estimation of the gene trees is under like maximum likelihood or whatever. Remember the first part of this talk. So you get gene trees, and then you combine the gene trees. And I've got this picture of Siavash Mirarab, who is a former student who developed ASTRAL. ASTRAL has become probably the basic method for doing species tree estimation. So ASTRAL is solving an NP-hard optimization problem, but in a constrained search space. So what is the NP-hard optimization problem? You have all of your gene trees. You encode them as quartet trees, unrooted trees on 4 leaves. And then you look for the species tree that maximizes the total number of quartet trees it agrees with in the gene trees. It's an NP-hard problem. If you solve it exactly, this is a statistically consistent method for species tree estimation under the multi-species coalescent model. Now, what they do is we constrain the search space by saying you're only allowed to reconstruct a tree whose bipartitions come from a certain set. That set contains the gene trees. Okay? The bipartitions in the gene trees are what you're allowed to use. In some cases, you might get additional ones. That constrained search space makes this problem polynomial time and does not change statistical consistency. It's used all over the place now. But statistical consistency is not so simple. The proof of statistical consistency assumes true gene trees. You do not have true gene trees. And a big part of the argument in the biology research community, the systematics research community, has been, should you use concatenation or should you use these summary methods? The summary methods assume true gene trees. The concatenation method may be inconsistent, but it gives results people like. So there's this big back and forth about this. But you just have to understand that the statistical guarantee of consistency depends upon true gene trees. You never get true gene trees. Gene trees always have error. Sometimes the error is very high. You can estimate how much error there is by looking at bootstrap support. Bootstrap support is often low in most genes, so especially when you're looking at genome-scale data. So how do methods perform when you have gene tree estimation error? So here's a simulation. This was done by a former student, Erin Molloy, published in Systematic Biology. What I want you to see is everything here is 26 species, but 1,000 genes. And we've taken the simulations with different sequence lengths, and we've binned the data sets by how much gene tree estimation error there is on average. So the 20 to 50%, that means not that bad. That means on average, the gene trees have between 20% error and 50% error. And then you have 50 to 80% error, and then you have 80 to 85% error. So you have high error in some cases. Now, I wouldn't think you should think of this as ridiculously high. It's very high. This is probably more normal. The middle one is probably way closer to normal. In the avian project, the average bootstrap support was 25%. If the bootstrap support is 25%, the error is probably 75%. So it's probably in here, okay? I just want you to realize estimation error is real. So on the y-axis is the species tree error. Remember, it's the percentage of the biopartitions that are wrong. So what am I showing? I'm showing 3 different summary methods: ASTRAL, which is a distance-based method, and MPS, which is maximum pseudo-likelihood. Then in red, I have SVD quartets, which is a method that is statistically consistent, but it's based upon looking at site patterns. And then concatenation using maximum likelihood, CAML. By the way, ML in this world means maximum likelihood. It does not mean machine learning. So just remember that. What do you see? For the low error, summary methods are the best. Low gene tree estimation error, summary methods give the best results. Concatenation is the next best, and SVD quartets is the worst. In the middle range, suddenly, concatenation is better. On the high range, concatenation is much better. And now SVD quartets is also good. What's really interesting here is that this is a high ILS condition. There's something called the anomaly zone in this research area. This is the anomaly zone. This is when the most probable quartet tree is different from the gene tree. Even in the anomaly zone, concatenation can be more accurate if there is enough gene tree estimation error. So for those of you who are ever going to do a species tree estimation, just bear in mind the relative accuracy of methods depends upon the data. Do you have a lot of ILS? Do you have a lot of gene tree estimation error? You're going to have to think about that when you think about methods, okay? Any questions about this? Okay. So back to the 1,000-plant transcriptome project, because now I'm switching gears. In the 1,000-plant transcriptome project, we're working with plants, lots of multicopy gene families, and we had to eliminate the multicopy gene families to do the analysis. We had 9,500 different gene families. Only 400 of them were single copy. We threw out 9,000 gene families, and we only had 400 left, and we did the phylogeny based upon what was left. Why did we do that? We did that because we didn't have methods to work with multicopy gene families. But let me just talk to you about multicopy gene families. This is an example where you have a 3-leaf species tree, a single gene evolving down this species tree, and it's evolving with a duplication at the root, and then it just continues to evolve. The gene family tree looks like 2 copies of the species tree, okay? If you had the gene family tree, correct, that's what it would look like. So you can have multiple copies of the leaves, and these are called multrees. Multi-copy trees, multrees, also called gene family trees. The problem is given a collection of these multrees to estimate the species tree. So this is saying, suppose you're not going to try to figure out orthology, you're just going to take the gene families, you're going to get trees on the gene families and try to combine them. So here's the species tree. Here's a gene family tree with 1 duplication, and it's a duplication here. Here's 1 duplication but 2 losses. So if you look, species A appears twice, B appears once, C appears once, D appears once. Okay? You might be able to figure out from this, maybe, that it looks like this, maybe not. But what about this one? You've actually lost 3 copies, and now you have only 1 copy of A, 1 copy of C, 1 copy of B, and 1 copy of D. So you think it's a single copy gene family, and you might think, okay, that's the species tree, but it's not. The copies that have lost are now giving you the wrong signal. So how do you get from a family, a collection of multrees, okay? So you can throw out multi-copy genes. That's what we did in the plant transcriptome project. You can try to figure out orthology, but that is not solved. Or you can run methods that combine gene family trees. There's various methods that do this. Some of them are based upon minimizing duplications and losses. Others do other things, okay? So 3 basic ideas, and we're interested in this 3rd one. Remember statistical consistency. We're interested in what methods can we prove statistically consistent. The first proof of anything being statistically consistent was from 2019, where we proved that the most probable quartet tree for if you take 4 species, the most probable quartet tree is the species tree, and therefore ASTRAL in the multi-individual context can be used as a statistically consistent method for species tree estimation from multrees. a quartet-based method being used because the most probable quartet tree is the species tree. So ASTRAL multi is statistically consistent, and it runs in polynomial time. So when Siavash Mirarab saw that we had done this, he said, that's interesting, but it's not even designed for this case, so why don't I work on something that's explicitly designed for this case? And he came up with something called ASTRAL Pro. And ASTRAL Pro is better in practice than ASTRAL multi, even though, as I'm going to show, it's not provably statistically consistent. So you have this 3 steps. In the first step, you take these multi-copy gene family trees and you root them according to your best guess at gene duplications and losses. And after you root them, you can identify each node as either a duplication or a speciation. And after you do that, you can say which are the quartet trees that you want to count. Okay, so these 3 steps starts with rooting every gene family tree, and then figuring out which nodes are duplications and losses and which quartets you're going to consider. If the rooting is done correctly, and if the gene family trees are correct, then this is statistically consistent. But that's a big if, because if you can do the rooting, then you've actually figured out orthology. So again, there's an if there, conditionally consistent. So what my student James and I worked on together was a different technique. We said, let's take these multi-copy gene family trees and cut them up so that they become single copy trees. And then we can feed those single copy trees to anything we like. And here are some results. So this is error. And we're comparing ASTRAL Pro to this technique of cutting things up and then running it through ASTRID, to the technique of cutting things up and running it through concatenation using maximum likelihood. And then this internal one is species RAX, which was developed by Alexis Stamatakis. So this is a condition with 101 species with both GDL and ILS. And on the x-axis is the number of gene family trees. As the number of gene family trees increases, error goes down for all methods. If you look carefully, you can see some differences between methods, but they're not big ones. In general, concatenation is very good. That's CA disco, by the way. That's breaking things up into pieces and then concatenating and then running maximum likelihood. CA disco doesn't appear here because you can't run RAXML on such a big data set. Runtime is really, there's differences though, because CA disco is very expensive, whereas ASTRAL disco is very fast. So ASTRAL disco is very fast. It's a little bit better than ASTRAL Pro, and it has even lower memory usage. Okay, 101 species, 1,000 species, 1,000 gene families, and now we're only comparing ASTRAL Pro, ASTRAL disco, and species RAX, because we can't run CA disco. Accuracy, these two are very close. They're both better than species RAX. Slight advantage to ASTRAL disco. Runtime, big advantage to ASTRAL disco. Big advantage for memory usage. So this technique, this disco technique, allows you to take anything on multi-copy gene family trees, cut the multi-copy gene family trees into single copy, and do what you want with it. Okay? Okay, summary of the species tree stuff is that for ILS-based species tree estimation, you do get statistical consistency, you do get fast methods, and these are much, much, much faster than concatenation. Remember the Avian project, 250 CPU years. If you just got gene trees and combined them, it's, I don't remember, right now it's probably a matter of just a day. I'm not sure how long it takes. You just get the gene family trees, you can combine them, it's very fast. Gene family trees, parallelized. For GDL, we have advances, but there's still a lot more to do. ASTRAL multi is conditionally statistically consistent. You need true rooted gene trees. Actually, ASTRAL multi is fine, but you do need true unrooted gene family trees. ASTRAL Pro, statistically consistent if you have true rooted gene family trees. CA disco and ASTRAL disco, no proof of statistical consistency, but good performance in practice. I did not show results with horizontal gene transfer. It turns out, though, that under a random model of HGT, Sebastian Roch has proven that the most probable quartet tree is still the species tree if you don't have too much HGT. And by species tree, I mean the underlying species tree on top of which you have HGT. And it turns out quartet methods like ASTRAL also give good accuracy. So there's this general thing. Quartet-based methods are actually really useful. They have provable guarantees, they reduce runtime, and they can be statistically consistent. So there's this nice thing about quartet-based methods in particular, okay? Okay. I think I have a little time, maybe 5 minutes? 5 minutes?
Yeah, a little more.
Okay, I'm going to quickly do some new stuff for improving scalability. This is stuff that probably no one here knows. I don't think anyone here knows. Okay, so remember DCM neighbor joining versus maximum likelihood, and we said it wasn't as good as maximum likelihood. Divide and conquer methods where you take a data set, you divide into subsets, you get trees on the subset, and you combine. The usual context is supertrees. You want the subsets to be overlapping so that their trees are overlapping. The problem with supertree methods, even though they're very well studied, they just don't scale. They really are not able to go to large data sets. Everything is basically an NP-hard optimization problem, and the heuristics don't work on large data sets. In the last few years, I've been working on a new technique called disjoint tree mergers. And this was developed by Molloy, who is a former student of mine. The idea is you take a data set, you divide into disjoint sets. You get trees on the disjoint sets, and then you combine them. Now, how do you combine disjoint trees? You need some auxiliary information. Erin's technique was to use a distance matrix she estimates to combine the trees. But when you're getting trees on the subsets, you don't have to do distance-based estimation. You do anything you like. And the key understanding here is that the disjoint sets, the trees there, you can't change them. But you allow the trees to blend. As long as you don't change them, they can blend. And so an example of blending is this second tree. These 3 trees are all subtrees of these 2 supertrees. But this one is a blended version. It's not like the red leaves are together, the blue leaves are together, and then the black leaves are together, okay? They intersperse. So as long as you force things to stay the same on the subset trees, the constraint trees, it's fine. And there's been a sequence of methods. Erin did the first one, NJ merge, then she improved it to tree merge. And then Satish Rao and I and one of his students developed something called constrained inc. Inc means incremental. And then finally, a student of mine, Vlad Smirnov, developed something called the guide tree merger. Each of these methods has provable guarantees in terms of statistical consistency. But what's really bizarre is that the guide tree merger is good. Now, why do I find it bizarre? It's the method that doesn't allow any blending at all. So you maintain statistical consistency. You can combine these pipelines with any method you like and any disjoint tree merger you like. The guide tree merger is the one that's working really well. And the open problem is to get a better method, okay? So the guide tree merger says you have these disjoint trees. You want to combine them to minimize the distance to an initial tree. If you allow blending, it's an NP-hard problem. If you do not allow blending, it's polynomial. By not allowing blending, I mean you just add edges between the subtrees. No blending allowed, polynomial. He implemented it. He tried to tell me it was going to work. I didn't believe him because blending is necessary, right? Well, guess what? It's really good. This is really bizarre. This is a result of combining that with ASTRAL. So ASTRAL on the subset trees, and you combine them using a distance-based beginning tree. So this, again, is error. And what we have here is what you get on 10 genes, 25 genes, and 1,000 genes. We're comparing the GTM ASTRAL here. Here's ASTRAL. Here's ASTRID. Here's NGST. It's more accurate than ASTRAL. It's using ASTRAL on subset trees, and it's more accurate. As you increase the number of genes, it maintains its advantage for accuracy. When you get to 1,000 genes, it's no longer really more accurate. But the fact that it's more accurate is really interesting, and it has to do with how ASTRAL constrains its search space. Look at the runtime. For 1,000 genes, the difference is 8,000 seconds for the pipeline, and 153,000 seconds for ASTRAL. It's a really big improvement in runtime, an improvement in accuracy, and you maintain statistical consistency. So what about scaling maximum likelihood? We're also getting advantages here, but now what we're doing is maximum likelihood on the subsets. And this is just a quick example with, again, error rate on simulated data. This is FastTree, which is fast method. This is IQTree. This is RAxML, and this is the pipeline. A simulated data with 1,000 sequences, RAxML is best. With another 1,000 sequences, but now it's not IID, it has selection going on. They're all about the same. Heterotachy with about 2,000 sequences. FastTree is not so great. The other methods are close. RAxML is a little bit better. RNA-Sim with 10,000 sequences or 50,000 sequences, and all of a sudden we see a big difference. Now the pipeline has the best accuracy. And in fact, the pipeline can run on 50,000, and RAxML does not give results. And in fact, it's not quite true. RAxML does give results, but it has essentially 100% error. So RAxML is doing a heuristic search. Its heuristic search is not suited to very large data sets. This divide and conquer allows you to do this. So this is generic use of disjoint tree mergers. The overall summary is large-scale phylogenomic estimation is really becoming feasible. There is a lot of reason to think about divide and conquer. It addresses heterogeneity across the genome. It addresses heterogeneity across the tree. And it allows you to take statistically consistent methods that are very computationally intensive and run them on subsets. You can't run them on the full data set. So the main open problems, empirical. Get a better divide and conquer pipeline. Do something better than GTM. It's got to be possible. The guide tree merger doesn't allow blending. If you allow blending, you should be able to improve accuracy. Theoretical questions. Are any of these pipelines absolute fast converging? I don't know. So for the mathematicians out there, nice question. Can we bound the error based upon the input? Can we develop a theoretical framework for why we sometimes improve accuracy? I think so. OK, so thank you very much. I'll be glad to talk to you later.